Imagine looking down the optics of your microscope at a day 3 embryo.
Right.
You know, you are staring at a gorgeous, morphologically flawless 8-cell. The blastomeres are perfectly symmetrical. There is like zero fragmentation.
The absolute textbook ideal.
Exactly. The textbook criteria, everything you have been trained to look for tells you this embryo is thriving. But molecularly, it might already be dead. It could be a biological ghost, just sort of running on fumes from an emergency stockpile.
Utterly incapable of taking the next developmental step, yeah.
Right. Welcome to this deep dive. For those of you listening who spend your days in the IVF lab, especially those of you preparing for your ESHRE clinical embryologist certification, we are about to explore the terrifying, beautiful reality behind that microscope lens.
It really is terrifying when you look closely at it.
It is. We are tracing the grand trajectory of early development, from the massive hoarding of maternal stores all the way to the ultimate biological handover, which is zygotic genome activation, or ZGA.
And we are talking about the most vulnerable, precarious phase in all of human biology here.
Without a doubt.
For the first few days of its existence, that embryo is essentially flying blind. It's executing a pre-written molecular script with absolutely no ability to rewrite the code if something goes wrong.
I always think of it like a molecular orchestra, but one where the composer died before the performance started.
Oh, that's a great way to put it.
Right. The sheet music is distributed, the instruments are tuned, and the cell is just praying every single section hits their cues.
Yeah, because if the string section comes in too early or the brass misses a beat, the whole symphony doesn't just sound bad.
It stops.
It permanently stops. The embryo arrests. And you, as a clinical embryologist, are sitting there trying to figure out why a morphologically perfect embryo just hit a brick wall.
Which is exactly why we need to unpack the biological reality of the oocyte today. Because the central dogma of biology, you know, DNA to RNA to protein, is effectively paused here.
Entirely paused. By the end of its growth phase, the oocyte has completely shut down active transcription.
RNA polymerase II is just silenced.
Right. So when that oocyte enters fertilization, it relies entirely on a finite, pre-calculated, pre-stored pool of messenger RNAs and proteins.
It is like a nuclear submarine that has just severed all communication with command and initiated a deep dive. There's no resupply.
Not at all.
The oocyte is, well, it's an expertly stocked emergency kitchen. Every single ingredient required to drive meiotic maturation, to survive fertilization, and to push through those first rapid cleavage divisions is already sealed inside.
You can't just run to the store for more flour.
Exactly. You have to selectively translate the maternal mRNAs you brought with you.
And that selective translation is entirely governed by proteostasis.
Which is such a delicate balance.
It's incredibly delicate.
Yeah.
It's the equilibrium of synthesizing new proteins from that stored mRNA, making sure those proteins fold into the correct three-dimensional shapes, and frankly, ruthlessly degrading the ones that are no longer needed.
Because ZGA at the 8-cell stage in humans is the critical checkpoint, right?
Yes, that's where the embryo finally turns its own DNA back on.
Yeah.
But to even reach that checkpoint, the proteostasis machinery has to function flawlessly.
But the reality of the lab is that things aren't flawless. I mean, we are taking this exquisitely sensitive system out of the fallopian tube and putting it into a plastic dish.
We expose it to light, to pH shifts.
Temperature fluctuations, yeah.
Yeah.
We introduce oxidative stress. And that stress can totally overwhelm the proteostasis machinery.
It absolutely can. And suboptimal culture conditions don't usually cause immediate explosive cell death.
Right, it's more insidious.
Exactly. They cause subtle, cascading failures in protein folding and degradation. When the proteostasis machinery gets overwhelmed, maternal proteins aggregate, the cell cycle halts, and you get that unexplained embryonic arrest.
At the 2, 4, or 8 cell stage. Which brings us to the specific knowledge milestones we're going to master today. If you are preparing for the ESHRE exam, or frankly, if you just want to be a better embryologist, you need to understand the physical mechanics of this vulnerability.
You really do.
So we are going to explore the ribosomal machinery and the exact phases of translation. We're going to dive deep into how the oocyte uniquely controls its translation, how it executes the maternal to zygotic transition, the mechanics of amino acid catabolism, and finally, how proteostasis dictates developmental competence in an RT setting.
That's a huge roadmap.
It is, but I have to ask you first. For someone staring at embryos all day, why do these specific molecular pathways matter so much?
Well, They matter because they are the invisible mechanisms driving the visible phenotype.
Ooh, I like that.
Yeah, you cannot understand why an embryo fails to activate its zygotic genome if you don't first understand how it was actively repressing its maternal genome.
That makes perfect sense.
And you can't appreciate the catastrophe of oxidative stress if you don't understand how proteins are physically built and folded. The ESHRE exam doesn't just test your ability to memorize facts, it tests your ability to connect a molecular disruption to a clinical outcome.
Right, so let's trace this journey logically. We'll start with the raw materials, the building blocks themselves.
The proteins.
Exactly. Then we'll look at the factories that assemble them. From there, we'll examine the master blueprints, how the oocyte hoards and selectively uses its maternal mRNA.
And finally, we will break down the maternal to zygotic transition.
Where the embryo attempts to change its entire operating system while the engine is still running.
That is the perfect progression, from the fundamental chemistry straight to the clinical reality.
Okay, let's start with the building blocks. The protein structure hierarchy.
Right.
We have 20 standard amino acids, and they organize into 4 levels of architecture. Primary structure is just the linear sequence of amino acids, like letters in a sentence.
Simple enough.
Yeah. Secondary structure is the local folding, the alpha helices and beta-pleated sheets driven by hydrogen bonds. Tertiary structure is the complete three-dimensional folding of the entire polypeptide chain.
And quaternary is when multiple folded subunits assemble together.
Like the multiple subunits of a DNA polymerase enzyme. But here is the trap, right? We tend to think of proteins as solid, indestructible little machines.
Which they aren't.
Exactly. Why is that tertiary structure, that 3D fold, actually so incredibly fragile in the early embryo?
Because that 3D fold is not locked in place by strong, unbreakable covalent bonds.
Oh, interesting.
Tertiary structure is largely held together by relatively weak interactions. We're talking hydrophobic collapse, hydrogen bonds, van der Waals forces.
So it's delicate.
Very. The protein folds specifically because its hydrophobic, water-fearing amino acids are trying to hide from the aqueous environment of the cytoplasm.
While the hydrophilic ones face outward.
Exactly. That creates a very specific but very precarious geometry.
And function is entirely dependent on that geometry.
Entirely. The active site of an enzyme, the highly specific binding pocket of a receptor on the cell surface, those only exist because the protein is folded precisely.
So if a reactive oxygen species, an ROS molecule from your culture media, manages to oxidize just 1 or 2 vulnerable amino acids on the surface of that protein.
The delicate balance of weak forces is disrupted. The protein begins to unravel. It loses its three-dimensional shape, and therefore it instantly loses its biological function.
Right, but in a normal somatic cell, say a skin cell or a liver cell, if a protein misfolds, it's not a catastrophic event, is it?
No. The cell just shreds the broken protein, goes into the nucleus, transcribes a new piece of mRNA, and prints a replacement.
Exactly, but remember our fundamental constraint here. The early embryo cannot do that.
Right. Prior to ZGA, transcription is silent.
It cannot go back to the DNA well to replace a damaged protein.
Which means the maternal pool of any given protein is a strictly finite, non-renewable resource.
That is so key for people to understand.
If an oxidative insult sweeps through the cytoplasm and misfolds 20% of a critical cell cycle regulatory protein, that 20% is gone forever.
Wow.
The embryo just has to survive on what is left until it can activate its own genome at the 8-cell stage.
That makes the physical architecture of these proteins arguably the most valuable currency in the lab.
Absolutely.
And it also means that the embryo isn't just a static entity, right? It is a highly active consuming metabolic engine.
Yes, it's constantly working.
Which brings us to amino acid catabolism. The embryo has to break down amino acids, not just to clear out the old ones, but to extract energy and raw materials.
Right.
And we know that measuring amino acid turnover in spent embryo culture media isn't just an academic exercise anymore. It's being actively studied as a non-invasive readout of viability.
And it makes perfect sense that it would be. The way an embryo metabolizes its amino acids tells you a story about its internal state.
But catabolizing an amino acid is complicated.
Very, primarily because of the nitrogen atom. You can't just drop an intact amino acid into the TCA cycle to make energy. The amino group has to be stripped off first.
And how does the embryo actually handle that?
Usually through transamination, the embryo transfers the amino group from the amino acid onto a molecule like alpha-ketoglutarate, which turns it into glutamate.
Ah, so that safely parks the nitrogen.
Yes. Alternatively, it can use oxidative deamination, which physically removes the amino group and releases it as a free ammonium ion.
But wait, free ammonium is highly toxic to cells, isn't it?
Extremely toxic.
In an adult human, the liver handles that by running the urea cycle to safely excrete it. An embryo sitting in a 20 microliter drop of culture media doesn't have a liver.
Which is a massive clinical vulnerability. If ammonium builds up in that microdrop, it will alter intracellular pH, disrupt metabolism, and impair embryonic development.
Which is exactly why modern media formulations are so obsessed with stability.
Yes. Glutamine, for instance, spontaneously breaks down into toxic ammonium at incubator temperatures.
Oh, right.
So we now use stable-type peptides like alanyl-glutamine to prevent that spontaneous breakdown. It protects the embryo from basically stewing in its own toxic waste.
That is such a direct link between the biochemistry and the daily lab routine.
It really is.
Okay, so once the nitrogen is safely stripped away, you're left with the carbon skeleton. And depending on what that specific skeleton looks like, the amino acid is categorized as either glucogenic or ketogenic.
Correct. If the carbon skeleton can be converted into intermediates of the TCA cycle, like pyruvate or oxaloacetate, it is glucogenic.
So amino acids like alanine or aspartate fall into this category.
Yes, the cell can literally use them to synthesize new glucose if needed. On the other hand, ketogenic amino acids, like lysine and leucine, break down directly into acetyl-CoA or acetoacetate.
So they can't be used to make glucose.
No, they are burned for energy or used to synthesize lipids. And a few, like isoleucine, are massive enough that they can be cleaved to feed into both pathways.
Okay, let's zero in on the ketogenic side for a minute. Specifically, the branched chain amino acids, or BCAAs. We are talking about leucine, isoleucine, and valine.
Right.
Now, in adult biology, BCAAs are famously metabolized in muscle tissue, but in our context, they're uniquely catabolized right inside the mitochondrial matrix of the oocyte and embryo.
And the spatial localization is key here. The oocyte relies heavily on oxidative phosphorylation. It needs robust mitochondrial function.
So by catabolizing these branched chain amino acids directly in the mitochondrial matrix?
The embryo efficiently generates NADH and FADH2 exactly where they are needed to drive the electron transport chain and produce ATP.
That is so efficient. But here is where leucine completely breaks the mold, right? Leucine isn't just a passive fuel source or a structural brick. It is actively pulling the levers of cellular growth.
Yes, leucine is a profound signaling molecule.
How so?
Well, when intracellular leucine levels are high, it acts as a potent activator of mTORC1, the mechanistic target of rapamycin complex 1.
The master regulator of cellular anabolism.
Exactly. When leucine binds to its sensors and activates mTORC1, it is essentially screaming at the cell, "Resources are abundant, stop recycling, start building, ramp up protein synthesis."
But what happens when the opposite is true? What if the embryo is experiencing amino acid deprivation? It can't just sit there and starve.
No, it initiates a desperate, highly coordinated survival response.
Yeah.
One of the most fascinating adaptations is the formation of mitochondrial-derived compartments, or MDCs.
NDCs?
Yeah. Under amino acid stress, the mitochondria will literally bud off these transient subcompartments.
Wow, just bud them off.
Yeah. These MDCs specialize in concentrating the machinery needed for amino acid degradation. They are trying to wring every last drop of usable energy and raw material out of the available pool.
While simultaneously sequestering damaged proteins to protect the rest of the organelle, right?
Exactly.
It's like a factory building a temporary emergency wing just to handle a sudden supply chain collapse.
That's a perfect analogy. And if that isn't enough, the cell will upregulate autophagy. It uses lysosomes to deliberately digest its own less critical cytoplasmic contents.
Like organelles and bulk proteins.
Just to recycle their amino acids back into the pool. It also ramps up the proteasome, identifying and shredding non-essential proteins. The embryo will literally cannibalize itself to ensure it has the critical amino acids needed to reach ZGA.
Which brings us to the actual design of embryo culture media. We don't just dump all 20 amino acids into a dish on day one. There is a deep metabolic logic to how we feed them.
Oh, absolutely. We provide non-essential amino acids early on, during the cleavage stages.
Why specifically the non-essential ones early on?
The embryo uses these non-essential amino acids primarily as intracellular osmolytes to regulate volume, as antioxidants to fight ROS, and as safe nitrogen buffers.
Okay, but as the embryo approaches the morula and blastocyst stages, its metabolic demands shift drastically.
Because ZGA has occurred. Transcription is roaring, and the embryo needs to build massive amounts of new tissue. That is when essential amino acids, like our MTO or activating leucine, become absolutely critical to support that biosynthetic explosion.
So you aren't just feeding the embryo, you are deliberately communicating with its metabolic sensors based on its developmental timeline.
Exactly.
Now, we've talked about the amino acid building blocks, and we've talked about the signaling to start building. Let's look at the factory floor itself, the protein synthesis machinery.
It's a ribosome.
If the amino acids are the raw materials, the ribosome is the ultimate 3D printer.
And what a machine it is. We are dealing with the 80S eukaryotic ribosome. It's composed of two distinct parts, a 40S small subunit and a 60S large subunit.
Okay, break down what they do.
The small 40S subunit is the reader. Its job is to bind to the messenger RNA and decode the sequence.
And the large one.
The large 60S subunit is the builder. It catalyzes the actual formation of the peptide bonds between the amino acids.
And the part that blows my mind every time I think about it is the peptidyl-transferase center, the actual catalytic core of the 60S subunit that links the amino acids together. It isn't a protein. It's a ribozyme.
Yes.
The chemical heavy lifting of creating the proteins that drive all life is actually performed by folded ribosomal RNA.
It is the ultimate echo of the RNA world hypothesis. Before proteins evolved to act as enzymes, RNA did the work.
It's a living fossil.
It really is. But before this magnificent 3D printer can do any work, it has to be built itself. And ribosome biogenesis is one of the most energy-intensive processes a cell can ever undertake.
Tell me about it.
It requires the coordinated transcription of ribosomal RNAs in the nucleolus, the import of dozens of ribosomal proteins from the cytoplasm, and their meticulous assembly.
And we know that in the oocyte, transcription shuts down. So when does this massive manufacturing effort happen?
It happens during the oocyte growth phase, deep in the ovary, weeks or months before the oocyte is ever ovulated.
Because it can't make them later.
Right, because the oocyte will have no ability to make new ribosomes during the critical window of early development. It must stockpile millions of them in advance.
And this biogenesis is heavily regulated by our old friend, the mTOR pathway.
Yes, it is.
Wait, let me stop you there. So if a growing oocyte in the follicle experiences nutrient stress, maybe the patient has severe metabolic issues and the mTOR pathway is suppressed, that oocyte might fail to build a large enough ribosomal stockpile.
That is the grim reality.
Yeah.
If that oocyte doesn't stockpile enough ribosomes during its growth phase, it might look perfectly normal upon retrieval.
It might even fertilize.
Exactly. But it will be fundamentally translationally compromised. It simply won't have the physical factory capacity to translate the maternal mRNAs needed to reach ZGA.
So you are watching a failure unfold on day 2 that was actually programmed into the oocyte months earlier by a supply chain failure.
Yes.
That is terrifying. It really recontextualizes the idea of oocyte competence.
It certainly does.
So assuming the oocyte successfully stockpiled its ribosomes, let's watch them work. Translation happens in 4 distinct phases: initiation, elongation, termination, and recycling. Let's break these down because the specific molecular players here are highly testable.
Okay, let's start with initiation. It's the most complex and the most regulated phase.
Yeah.
It begins with the assembly of the 43S pre-initiation complex.
Right.
You have the small 40S ribosomal subunit binding with several initiation factors. Crucially, you have EIF2, which is bound to a molecule of GTP, carrying the initiator methionine transfer RNA.
So we have the reader subunit, and it's holding the very first amino acid, methionine, ready to go.
Yes. This complex then has to find the mRNA. It relies on another set of factors sitting on the 5' end of the messenger RNA.
The cap.
Exactly. The key player here is EIF4E, which physically binds to the protective 5' cap of the mRNA. Another protein, EIF4G, acts as a massive scaffold, connecting the cap-binding proteins to the ribosome complex.
So they dock, and then the ribosome has to physically scan down the mRNA strand, burning energy looking for the start sequence, the AUG start codon.
Once it finds the AUG, the GTP on EI2 is hydrolyzed, the initiation factors fall away, and the large 60S subunit slams down into place.
And boom, you now have a fully assembled, functional 80S ribosome locked onto the start codon. Initiation is complete.
Correct. And now we move to elongation. The ribosome has 3 distinct chambers: the A site, the P site, and the E site.
Acceptor, peptidyl, and exit.
Right. The initiator methionine is sitting in the P site. The A site is empty, waiting for the next amino acid. A factor called EEF1A brings the next appropriate transfer RNA into the A site.
And if the anticodon matches the mRNA, our ribozyme core catalyzes a peptide bond, transferring the growing chain onto the new amino acid in the A site.
Yes, and here is where the mechanical strain really hits. The ribosome now has to shove everything forward to read the next codon.
That sounds energy intensive.
It is. That physical ratcheting is driven by elongation factor 2, or EEF2. It requires the hydrolysis of another GTP molecule to forcibly translocate the ribosome exactly 3 nucleotides down the mRNA strand.
So the empty tRNA moves to the E site and is ejected. The growing peptide chain is now in the P site, and the A site is empty again. Think about the massive energy drain there. The cell is burning GTP to load the tRNA and burning another GTP to ratchet the ribosome. Multiply that by a protein that is 1,000 amino acids long.
Millions of times over.
Multiply that by millions of proteins. If the embryo's mitochondria are struggling and ATP and GTP levels drop, elongation will literally stall.
The ribosome will just stop moving.
But if there is enough energy, it chugs along until it hits a stop codon, initiating termination.
Right. The stop codons UAA, UAG, and UGA do not code for an amino acid. There is no tRNA for them.
So what happened?
Instead, they're recognized by release factors, specifically ERF1 and ERF3. These release factors enter the A site and force the ribozyme to add a water molecule to the end of the peptide chain instead of an amino acid.
So this severs the connection and the newly synthesized completely linear protein floats away.
Yes, but the ADS ribosome is still clanked onto the mRNA. We need to recycle it.
That requires a specialized protein called ABCE1, right?
Exactly. It uses ATP to pry the large 60S and small 40S subunits apart. The mRNA is released, and the 2 ribosomal subunits are free to go initiate another round of translation.
Out of all these incredibly complex, highly choreographed steps, where does the cell actually exert control? Like, if an embryo wants to reduce the production of a specific protein, does it stop the ribosome halfway through elongation?
Never. That would be a massive waste of the ATP and GTP it already invested. The control is almost entirely at the initiation phase.
Initiation is the bottleneck.
It is the rate-limiting bottleneck.
Yeah.
If you want to stop a protein from being made, you prevent the 43S pre-initiation complex from ever docking with the mRNA cap.
As we will see when we discuss how the oocyte hoards its maternal stores, manipulating that cap-binding protein, EIF4E, is the primary mechanism of translational control.
Exactly.
But before we talk about how the oocyte silences these messages, we need to talk about where this translation is physically happening in the cell. Ribosome localization.
Right. Geography matters.
A ribosome isn't just a ribosome. We have free ribosomes floating in the cytosol, and we have ribosomes bound to the rough endoplasmic reticulum. The golden rule here is where a protein is made determines where it ultimately lives.
Yes, the cellular geography is highly specific. Free ribosomes synthesize proteins that are destined to remain in the cytosol, or perhaps travel to the nucleus or the mitochondria.
But if a protein needs to be secreted outside the cell, or embedded into a plasma membrane, or sequestered inside a lysosome,
it absolutely must be synthesized at the rough ER.
But how does a ribosome floating in the cytoplasm know that the specific mRNA it just grabbed codes for a secreted protein? How does it know it needs to move to the ER?
The mRNA itself contains a ZIP code. As the very first few amino acids, the N-terminus emerge from the ribosome during elongation, they form a specific signal peptide.
Okay.
As soon as that signal peptide peeks out of the ribosome's exit tunnel, it is recognized by a massive ribonucleoprotein complex called the signal recognition particle, or SRP.
So the SRP acts like a molecular crossing guard.
More like a molecular escort. The SRP physically grabs the signal peptide and immediately halts translation.
Oh, it just stops it right there.
It stops the ribosome from making any more of the protein. The SRP then drags the entire paused ribosome complex through the cytoplasm over to the membrane of the endoplasmic reticulum. It docks the ribosome onto a specialized pore in the ER membrane called a translocon.
Once it's securely docked, the SRP releases and translation resumes.
Right.
But now, instead of the protein spilling out into the cytoplasm, it is being spooled directly through the translocon pore into the inner lumen of the ER as it is being synthesized, co-translational translocation.
And once inside the protective environment of the ER lumen, the protein undergoes critical post-translational modifications. Signalopeptidases cleave off the targeting sequence.
Sugar chains are added too, right?
Yes, enzymes add complex sugar chains in a process called N-linked glycosylation. Disulfide bonds are formed to lock the 3D structure in place.
Let's connect this beautiful cell biology directly to what clinical embryologists see under the scope. Why do we care so deeply about rough ER synthesis in the oocyte?
Because of the zona pellucida.
Yes.
The zona pellucida is not a static shell. It is a complex extracellular matrix composed of heavily glycosylated proteins, primarily ZP1, ZP2, and ZP3.
And these zona proteins are synthesized entirely by the oocyte's rough ribosomes during the oocyte growth phase.
Exactly. They must be perfectly translocated into the heavily modified with sugars, and then secreted outside the cell to form that thick, protective layer.
And the zona pellucida is the absolute front line of reproductive security. It is essential for species-specific sperm binding.
Right.
More importantly, when the first sperm fuses with the oocyte, cortical granules undergo exocytosis and cleave ZP2, rapidly hardening the zona to block any other sperm from entering.
The block to polyspermy.
Exactly. If the oocyte's rough is stressed, or if there's a defect in cotranslational translocation or glycosylation, the zona pellucida will be structurally compromised. And a compromised zona in the IVF lab leads to disaster. You see abnormal fertilization, you see polyspermy, multiple sperm entering the oocyte, yielding highly abnormal, non-viable 3PN embryos.
A structural failure of the zona is fundamentally a failure of rough protein synthesis.
So we've synthesized the protein, we've put it in the right location, now we have to fold it.
The hardest part.
We already established that the linear string of amino acids is useless until it achieves its tertiary structure. The cytoplasm is incredibly crowded, chaotic, and full of interacting molecules. A protein trying to fold on its own would just tangle into a useless aggregate.
Which is why the cell employs molecular chaperones. These are specialized proteins whose sole job is to assist other proteins in achieving and maintaining their correct 3D shape.
Okay, so how do they work?
As soon as a nascent polypeptide chain begins emerging from the ribosome, chaperones like HSP70 and its co-chaperone HSP40 swarm it. They bind to the exposed hydrophobic regions of the chain.
To protect them.
Exactly, preventing those regions from inappropriately sticking to themselves or to other proteins while the rest of the chain is still being synthesized.
They act as shields, but there are different chaperones for different jobs, right?
Oh yeah. HSP90, for example, is highly specialized. It doesn't bind to every nascent protein. It specifically targets client proteins involved in cellular signaling.
Like kinases, steroid hormone receptors.
Yes, ensuring these critical communication nodes are perfectly folded. And inside the the master chaperone is BP, also known as GRP78. It oversees the folding of all those secreted proteins, like our zona pellucida glycoproteins.
But the most structurally fascinating chaperones are the chaperonins, specifically the HSP60 complex, also known as GRO-EL in bacteria.
They are incredible structures.
I am obsessed with HSP60. I think of it as a cellular safe room. It isn't just a protein that binds to a chain. It is an actual physical barrel-shaped chamber.
It really is.
If a protein is struggling to fold, or if it has been partially denatured by heat or oxidative stress, it gets placed completely inside the hollow cavity of this barrel. A lid, the HSP10 co-chaperonin, seals the top.
It creates a microenvironment that is completely isolated from the chaotic, crowded cytoplasm.
And then what happens inside?
Inside that safe room, the hydrophobic walls shift to hydrophilic, forcing the trapped protein to collapse inward and try to find its correct native thermodynamic fold.
And this entire process, opening the lid, changing the walls, it burns massive amounts of ATP. The cell pays a high energy premium to salvage a misfolded protein.
But chaperones aren't miracle workers.
Right. What happens when a protein is so heavily oxidized or mutated that it simply cannot be salvaged, even inside the HSP60 safe room?
Then the cell has to make a ruthless calculation. A severely misfolded protein is not just useless, it is highly dangerous.
Because of the exposed hydrophobic regions.
Exactly. They will stick to other proteins, forming toxic, insoluble aggregates that can physically disrupt cellular processes and trigger apoptosis. The cell must initiate protein quality control, or PQC, to destroy the threat.
And the primary weapon in the PQC arsenal is the Ubiquitin-Proteasome system, the UPS. How does the cell know which protein to destroy?
It uses a tagging system. Specialized enzymes identify the irreparably misfolded protein and covalently attach a chain of small regulatory proteins called ubiquitin to it.
A polyubiquitin chain.
Yes. This polyubiquitin chain acts as a highly specific molecular barcode. It screams, "Destroy me."
And that barcode directs the doomed protein to the 26S proteasome. If HSP60 is a safe room, the proteasome is a molecular wood chipper.
Very accurate.
It is a massive, barrel-shaped protease complex. The ubiquitinated protein is fed into the top, unfolded, and shoved down the central channel where the catalytic sites systematically shred it back into short peptide fragments.
Those peptides are then broken down further into individual amino acids to be recycled.
But the UPS can only handle individual proteins.
Right. If the stress is so severe that proteins have already clumped together into large aggregates, the proteasome can't fit them.
So what then?
The cell must then rely on lysosomal autophagy, enveloping the entire aggregate in a membrane and delivering it to a lysosome for bulk degradation.
Wow.
And if the misfolding occurs inside the endoplasmic reticulum, the cell uses a highly complex pathway called ERAD, ER-associated degradation.
ERD is wild. The protein is inside the ER, but the proteasome is out in the cytoplasm. So the cell literally has to unfold the defective protein and thread it backward, out through a pore in the ER membrane, back into the cytoplasm, ubiquitinate it on the fly, and feed it directly into the proteasome.
It shows the lengths the cell will go to prevent proteotoxic stress. Maintaining this proteostasis is a matter of life and death for the early embryo.
Yeah, if the folding machinery and then the degradation machinery get overwhelmed, the embryo is doomed.
Absolutely doomed.
Which brings us to a massive shift in our deep dive. We have established the basic rules of how cells build and manage proteins. Now we enter the unique, bizarre, and highly specialized world of translational control in reproductive biology.
We are leaving standard somatic cell biology behind here.
Yes, we are. This is where we see the true elegance of developmental programming. Let's go back to that massive reserve of maternal mRNAs built during oogenesis.
Right.
The germinal vesicle, or GV oocyte, is sitting in the ovary. It has completed its transcriptional phase. It is utterly silent.
Completely dormant transcriptionally.
It contains thousands of different mRNA transcripts, representing a vast pre-planned genetic program that must execute sequentially, step by step, all the way from meiotic resumption to zygotic genome activation at the 8-cell stage.
Right.
What are the key transcripts in this dormant stockpile that an embryologist absolutely must know?
The exam will almost certainly test your knowledge of these specific maternal stores. First, cyclin B. This is the regulatory subunit of MPF maturation promoting factor.
Okay, cyclin B.
The cyclical translation and degradation of cyclin B is what physically drives the oocyte, and later the embryo, through the meiotic and mitotic cell cycles.
Next is CMOS. I think of CMOS as the emergency parking brake.
Good way to remember it.
It is a kinase that is absolutely critical for establishing and maintaining the mature oocyte in metaphase M2 arrest. Without the continuous translation of CMOS, the oocyte wouldn't wait for a sperm.
It would just spontaneously activate.
Right. It would blow right past metaphase M2 and parthenogenetically divide, which is a dead end.
Exactly. Third on the list is CPEB, the cytoplasmic polyadenylation element binding protein. If the maternal mRNA pool is a locked library, CPEB is the head librarian.
Okay.
It is the master regulator that decides which mRNAs remain dormant and which ones get translated.
And finally, the spindle assembly checkpoint, or SAC proteins like BUB-R1 and MAD2.
The SAC proteins are the quality assurance inspectors. They ensure that all chromosomes are perfectly aligned on the metaphase plate before the cell is allowed to divide, preventing aneuploidy.
The sheer temporal duration of this maternal control is what makes it so staggering. These proteins are encoded by mRNAs that might have been transcribed months ago, yet they dictate the precise timing of chromosome segregation on day 2 of embryo culture.
It's incredible.
When a patient presents with poor oocyte quality and we see recurrent failure to reach the blastocyst stage, we are very often looking at a fundamental deficiency or degradation in this exact maternal stockpile.
Very frequently, yes.
Okay, so we have thousands of these vital mRNAs, cyclin B, CMOS, sitting in the cytoplasm of the GV oocyte. The cell is full of functional ribosomes. Why aren't they being translated right now?
What keeps them quiet?
Yeah, what physically keeps them quiet, and what is the key that finally unlocks them when the oocyte receives the signal to mature?
The silencing mechanism is incredibly robust. It relies on 2 physical barriers on the mRNA molecule itself. A severely truncated poly-A tail at the 3' end, and a complex of repressor proteins that block the 5' cap.
Walk us through the repressor proteins. How do they physically block the ribosome?
In the 3' untranslated region of these dormant maternal mRNAs, there is a highly conserved sequence of nucleotides called the CPE, the cytoplasmic polyadenylation element.
Okay, the CPE?
Our master regulator, CPEB, physically binds to this CPE sequence. When the oocyte is dormant, CPEB is in an unphosphorylated state. In this state, it recruits a repressor protein called maskin,
Maskin is the bouncer at the club.
a very aggressive bouncer. Maskin binds to CPEB at the 3' end, but then it reaches all the way across the mRNA molecule and binds directly to EIF4E, which, as we established earlier, is the critical initiation factor sitting on the 5' cap.
Wow, so by binding to EIF4E, maskin completely blocks the scaffold protein EIF4G from docking. The 43S pre-initiation complex simply cannot assemble.
Exactly. Maskin forces the mRNA into a physical closed-loop structure. The 5' and 3' ends are locked together. The mRNA is translationally dead. It is utterly invisible to the ribosomes.
And other proteins help out too.
Yes, other RNA-binding proteins like pumilio and musashi, as well as specific microRNAs, also pile onto the 3' end to help enforce this absolute silence.
So that is the lock. A closed loop maintained by maskin. What is the key? The patient receives an HCG trigger shot, the LH surge hits the follicle, the oocyte is told to resume meiosis. How does the oocyte instantly unlock these specific mRNAs without transcribing anything new?
It uses a massive kinase signaling cascade. When maturation is triggered, kinases like MPF become active. They rush into the cytoplasm and phosphorylate CPEB.
Just a single phosphate group.
That single addition of a phosphate group fundamentally changes the three-dimensional conformation of CPEB.
It changes shape. And when it changes shape, it kicks maskin to the curb.
Maskin loses its affinity and dissociates.
Right.
Suddenly, EIF4E on the 5' cap is exposed. EIF4G binds, the ribosome pre-initiation complex docks, and translation can theoretically begin. The lock is broken.
But simply removing the repressor isn't enough to drive the massive explosive burst of translation required for maturation.
No, it's not. To really turn up the volume, the oocyte utilizes the cytoplasmic polyadenylation switch.
I want to spend significant time on this switch because it is brilliant. When we learn basic biology, we are taught that the poly A tail is just a protective bumper added in the nucleus to stop the mRNA from degrading.
Right.
But in the oocyte, the length of the poly A tail is actually a volume dial.
That is the most accurate way to visualize it. It is a highly sensitive dimmer switch. A short poly A tail, say less than 20 adenines, means the mRNA is translationally silent.
And a long tail.
A long tail extended to 150 or more adenines means that mRNA is going to be translated at maximum velocity.
And the mechanical reason a long tail drives high translation is because of a protein called PAB, poly A binding protein.
Yes. A long poly A tail recruits dozens of PBP molecules.
Wow.
PBP powerfully interacts with the initiation factors at the 5' cap. It recircularizes the mRNA, but this time in an activating loop.
So it's a good loop this time.
Exactly. As soon as a ribosome finishes translating the mRNA and falls off the 3' end, the PABP loop immediately feeds it right back onto the 5' cap. The ribosomes just cycle continuously, massively amplifying protein production.
So walk us through the actual mechanism of how the oocyte physically extends that tail in the cytoplasm. We left off with CPEB being phosphorylated and kicking off maskin.
Right. At the time of germinal vesicle breakdown, Aurora A kinase acts as the critical trigger. It phosphorylates CPEB. This activated CPEB now acts as a beacon, recruiting a large protein complex called CPSF to the 3' end.
Okay, so CPSF is recruited.
And CPSF then recruits the actual enzyme that performs the extension, a specialized cytoplasmic polymerase called GLD2.
So GLD2 docks, and it just starts blindly adding adenine nucleotides, one after another, extending the tail from 20 to 150.
Exactly. As the tail grows, PABP coats it, the active loop forms, and the dormant mRNA suddenly erupts into translation. And the beauty of this system is that it works in reverse as well. Enzymes like PARN and the CCR4 knot complex can be recruited to aggressively chew the poly A tail back down to 20 nucleotides, instantly silencing the mRNA again.
Meaning the oocyte can choreograph a perfectly timed, highly complex dance of protein expression. It can extend the tail on cyclin B, driving the cell cycle forward. Then it can deannihilate cyclin B to allow chromosome segregation, while simultaneously polyadenylating CMOS to hold the oocyte at metaphase M2.
Yes.
All of this changing protein landscape occurs without a single new transcript being synthesized.
And the clinical implications of this dimmer switch are massive. Disruption of Aurora A kinase or mutations affecting CPEB directly result in catastrophic oocyte maturation failure.
Really?
Yeah. If you have a patient whose oocytes consistently arrest at the GV stage, or arrest at metaphase M1, despite a perfect HCG trigger and perfect lab conditions, you're very likely looking at a fundamental failure of this specific polyadenylation switch.
Wow.
The meiotic spindles simply cannot assemble because the necessary spindle proteins never had their poly A tails extended.
Wow. You are staring at an M1 arrest, and the root cause is a broken cytoplasmic polymerase.
It's all connected.
Okay, so we've activated the maternal software. The oocyte has matured, it has ovulated, and it has been successfully fertilized by a sperm. The embryo now wants to start its own life. It wants to boot up its own zygotic genome.
Right.
But here is the critical vulnerability. You cannot install the embryo's new operating system without aggressively, actively uninstalling the maternal one, maternal mRNA clearance.
This is a point that is so frequently misunderstood in embryology. Maternal mRNA doesn't just passively fade away over time. It is actively hunted down and destroyed.
Effectively hunted.
Yes. If the embryo fails to clear those old maternal messages, it will attempt to execute two entirely conflicting developmental programs at the exact same time. The cell cycle will derail, and the embryo will arrest.
Wait, so if the oocyte is actively destroying its own mRNA, the very instructions it relied on to survive this far, isn't that biological suicide?
It sounds counterintuitive, but it is absolute self-preservation. Think about CMOS. Its entire job is to halt the cell cycle and arrest the oocyte at metaphase M2.
Okay.
Once fertilization occurs, if CMOS isn't immediately destroyed, the embryo can never undergo its first cleavage division. The parking brake is still pulled.
So how does the purge happen? What are the mechanics of the uninstallation?
In mammals, this massive degradation occurs in two highly coordinated waves. Wave 1 is entirely oocyte-driven. It utilizes maternal degradation machinery and begins immediately upon fertilization.
And wave 2?
Wave 2 is embryo-driven and coincides perfectly with zygotic genome activation, meaning the new embryonic genome actually transcribes the executioners to finish off the remaining maternal transcripts.
The mechanisms of destruction are brutal. Walk us through them.
The primary mechanism is deadnylation-dependent decay. Our old friend, the CCR4 knot complex, is recruited to the target mRNA. It aggressively removes the entire poly-A tail,
Which destabilizes it.
once the tail is gone, the mRNA is highly unstable. An exonuclease called XRN1 then binds to the exposed end and rapidly chews the mRNA up into individual nucleotides.
There's also endonucleolytic cleavage, where the RASC complex, guided by highly specific microRNAs, acts like molecular scissors and just slices the mRNA right in the middle, destroying the coding sequence.
Yep.
And finally, decapping enzymes physically lop off the protective 5' cap, leaving the mRNA utterly defenseless against background nucleases.
The clinical cutoffs here dictate success or failure. Approximately 30% of all maternal transcripts must be destroyed within the first 24 hours post-fertilization.
30%.
If the denylation machinery fails to clear most in cyclin B1, the cytostatic factor remains active, and the embryo cannot exit metaphase M2 arrest.
If an embryologist is looking at their time-lapse incubator, what is the visual phenotype of a failure in wave 1 clearance?
They will see recurrent developmental arrest at the 2-cell or 4-cell stage. The blastomeres might look healthy, but they simply refuse to cleave again. The maternal program is hanging on, stubbornly preventing the mitotic cell cycle from progressing.
Which perfectly sets up the ultimate hurdle, the focus of Section 4 of our deep dive, the maternal to zygotic transition, or MZT. This is the grand handoff.
It is.
MZT is not a single event. It is two simultaneous, overlapping processes. It is the maternal mRNA clearance we just discussed, happening concurrently with zygotic genome activation. Let's get the absolute specific timing straight, because the difference between human and mouse models is a classic trap that catches embryologists on exams and in the lab.
It is a critical distinction. In human embryos, minor ZGA begins at the 4-cell stage. This is a very small, restricted wave of transcription, almost like the embryo testing the waters, transcribing a few essential regulatory genes.
But major ZGA.
Major ZGA, the massive global burst of embryonic transcription where RNA polymerase thyrsin fully re-engages and takes over control of development, happens at the 8-cell stage.
And in mice.
In the mouse model, which so much of our foundational research is based on, major ZGA happens much earlier, at the 2-cell stage.
This timing difference in humans is exactly why clinical embryology is so deceptively tricky. Let's go back to my hook from the beginning of the show. You have an embryo that looks morphologically perfect on day 3. A beautiful, symmetrical 8-cell embryo. How can it be molecularly doomed?
Because morphology on day 3 only tells you one thing. It tells you that the maternal stockpile of proteins and mRNAs was sufficient to drive those first few cleavage divisions.
Right.
It tells you absolutely nothing about whether the new embryonic genome has successfully activated. If the massive epigenetic hurdles of major CGA fail at that 8-cell stage, the embryo will look fine today, but it will rest right there or perhaps at the morula stage tomorrow.
Because it's out of supplies.
It has no functioning mRNA to produce the new proteins required to form a blastocyst.
So what does it actually take to successfully activate the embryonic genome? It's not just a matter of transcription factors binding to a promoter. You have to physically unpack the DNA. We need to talk about chromatin remodeling at ZGA because the physical task of turning incredibly dense, silent paternal sperm chromatin into open to tipitant embryonic chromatin is staggering.
The scale of the epigenetic reprogramming required is difficult to overstate. Let's start with the paternal genome.
Okay.
The sperm DNA arrives highly compacted, packaged in protamines. Protamines are incredibly basic, positively charged proteins that pack the DNA so tightly it can fit inside the tiny sperm head.
It's like a crystal.
Effectively, yes. Within hours of fertilization, the oocyte must actively strip every single one of those protamines off the paternal DNA and replace them with maternal histones.
And even after you swap the packaging, the DNA is still heavily methylated. The embryo has to erase the parental epigenetic imprints to achieve a state of true totipotency.
Yes, the genomes must be blank slates. The paternal genome undergoes rapid active demethylation. Enzymes called TET proteins literally oxidize the methyl groups right off the cytosine bases.
What about the maternal genome?
The maternal genome, interestingly, undergoes passive dilution. It simply loses its methylation marks gradually as the cells divide and replicate their DNA without maintaining the marks.
But even when the DNA is demethylated and wrapped in histones, it is still physically too condensed for the massive machinery of RNA polymerase II to get in and transcribe it. When we hit the 8-cell stage, the chromatin has to be forcibly wrenched open.
This requires powerful chromatin remodeling complexes. First, histone acetyltransferases, or HATs, attach acetyl groups to the histone tails.
And what does that do?
This neutralizes the positive charge of the histones, drastically loosening their electrostatic grip on the negatively charged DNA backbone.
And then the heavy machinery moves in. ATP-dependent remodelers, like the SWES-SNF complex, these complexes use massive amounts of energy to literally slide the nucleosomes down the DNA strand or evict them entirely, physically exposing the promoter regions of the zygotic genes.
And simultaneously, the Ubiquitin-Proteasome system is working in absolute overdrive to seek out and degrade any lingering maternal repressor proteins that might be blocking those promoters.
Once the promoters are clear, the activating epigenetic marks appear, specifically H3K4ME3 and H3K27AC.
Yes.
Transcription factors bind, RNA polymerase II engages, and the zygotic genome roars to life. It is a massive, highly coordinated epigenetic renovation.
But this renovation is heavily dependent on cellular signaling, specifically the MEPK and PI3K pathways.
So if there's stress...
If the embryo is stressed, if its energy levels are low due to mitochondrial dysfunction, or if it's fighting off ROS in the culture media, this epigenetic renovation stalls. The Swiss-NSF complexes don't have the ATP to slide the nucleosomes. ZGA fails.
Which brings us perfectly to the linchpin of this entire precarious window: proteostasis and developmental competence. We said earlier that the embryo cannot transcribe new chaperones until ZGA, yet it faces an enormous, unrelenting proteostatic demand.
It really does.
It is rapidly dividing, it is constantly reorganizing its cytoskeleton, it is aggressively degrading maternal proteins, and it is trying to remodel its entire genome. All of this must be accomplished with a fixed, finite supply of folding and degradation machinery.
It has to be the most vulnerable window in mammalian biology. The UPS has to selectively and massively degrade cyclin B to allow exit from metaphase M2.
The heat shock proteins are working overtime,
trying to keep the newly translated maternal proteins from misfolding. Autophagy is attempting to clear out massive, aging maternal organelles like mitochondria. The unfolded protein response, or UPR, is constantly monitoring the ER to ensure the zona pellucida and cell surface receptors are folding correctly.
It is a proteostatic tightrope, and the consequences of falling off are catastrophic. If misfolded proteins aren't cleared immediately, they aggregate. These aggregates are fundamentally toxic. They physically gum up the cellular machinery.
And crucially, the meiotic and mitotic spindle apparatus is exquisitely sensitive to this proteotoxic stress.
Extremely sensitive.
Highly sensitive. If the tubulin proteins, or the SAC proteins, misfold, and the chaperones are too overwhelmed to fix them, the spindle assembles abnormally.
And then what?
When an abnormal spindle attempts to segregate the chromosomes during a cleavage division, chromosomes lag. They missegregate. The result is aneuploidy. A highly significant percentage of the chaotic aneuploidy we see in cleavage stage human embryos is directly driven by a breakdown in maternal proteostasis, not necessarily an inherent genetic error in the DNA.
And ultimately, if the ER stress becomes completely overwhelming, the unfolded protein response will switch from a rescue mission to an executioner, triggering apoptosis, and the embryo will shatter into fragments.
Design line.
Let's tie all of this deep molecular biology directly to the clinical reality of the IVF lab. The clinical significance of protein metabolism failures. We are constantly fighting three massive enemies: oocyte aging, oxidative stress, and the demands of extended culture. Let's start with aging. We know older patients have higher aneuploidy rates, but why, molecularly?
Oocyte aging, whether it is chronologically in the patient's ovary or in vitro if the oocyte sits too long in the dish before insemination, is fundamentally a loss of proteostasis.
Right.
As the oocyte ages, its pool of functional chaperones depletes. The efficiency of the 26S proteasome declines. The oocyte simply loses the physical capacity to clear its own waste or fold its proteins correctly.
So the spindle proteins misfold, the chromosomes missegregate, and viability plummets.
Exactly.
And then we subject these incredibly fragile aging cells to in vitro culture. And no matter how good our incubators are, we introduce oxidative stress, reactive oxygen species, or ROS.
ROS, or absolute wrecking balls to tertiary protein structure. They directly oxidize the side chains of vulnerable amino acids, particularly methionine and cysteine.
And when a cysteine residue is oxidized,
The critical disulfide bonds that hold the protein's 3D structure together collapse. The protein unravels. The cell desperately tries to throw chaperones at it. The UPS gets overwhelmed trying to clear the massive influx of garbage, and the entire proteostatic system crashes.
and finally, extended culture. We push embryos to day 5 or day 6 routinely now to select for the best blastocysts. But that strategy has a hidden cost.
Extended culture relies entirely, 100%, on a robust and highly successful zygotic genome activation.
It has to work.
If you push an embryo to day 5, but its ZGA was weak or delayed perhaps due to a slight epigenetic stalling at the 8-cell stage, that embryo will rapidly exhaust its remaining maternal protein stores long before the new zygotic proteins can take over the heavy lifting. It starves in the dish.
The clinical readouts of these specific failures are so clear once you understand the timeline. If you see recurrent 2 to 4 cell arrest, you are looking at a failure of minor ZGA or a failure of the CCR4 NOT complex to clear the maternal transcripts.
Yes.
If you see a massive drop-off in arrest at the 8-cell stage, that points squarely to a failure of major ZGA and chromatin remodeling. And what about 3PNs? A fertilization check reveals 3 pronuclei.
A 3PN typically means the blocked polyspermy failed. Multiple sperm entered. And we now know that the polyspermy block depends entirely on the correct rough ER synthesis of zona proteins and the rapid, perfectly timed translation of specific stored maternal mRNAs to trigger cortical granule exocytosis.
So a 3PN can very often be a direct, visible readout of a massive translational failure within the oocyte.
It absolutely can.
So when a lab director is choosing the right culture media, it isn't just about following the manufacturer's protocol or chasing the latest trend. It is a highly reasoned, molecularly calculated decision designed to support this exact precarious timeline.
This is the entire biochemical rationale behind sequential media, and why single-step media have to be so exquisitely balanced. You must provide non-essential amino acids early on.
For volume and ROS buffering.
The cleavage stage embryo uses them as intracellular buffers against that ROS damage and as osmolytes to maintain volume. Then you absolutely must ensure that essential amino acids, particularly our mTOR activator leucine, are available from the morula stage onward.
Because once major ZGA hits at the 8-cell stage, the embryo needs a massive influx of raw materials and anabolic signaling to build a blastocyst.
Exactly.
Okay, let's take a deep breath and summarize this incredible journey. For those of you preparing for the exam or just prepping for your shift tomorrow, here's the final synthesis. We started with the sheer vulnerability of the 3D protein fold, emphasizing that a misfolded maternal protein is a permanently lost resource. We covered the catabolism of amino acids, dealing with toxic ammonium, and highlighted leucine's unique role as the master switch for mTORC1 anabolism. We built the incredibly energy-hungry ADS ribosome, marveled at its RNA catalytic core, and identified initiation, specifically the binding of EAF4E to the cap, as the ultimate bottleneck for translational control.
We tracked those ribosomes over to the rough ER to synthesize the vital zona pellucida glycoproteins, the front line against polyspermy. We watched the HSP60 safe rooms and the 26S proteasome wood chippers battle to maintain the delicate balance of proteostasis.
We then unlock the oocyte's deepest secrets, the massive translationally silent maternal mRNA stockpile, the elegant dimmer switch of cytoplasmic polyadenylation, where Aurora A kinase and GLD2 lengthen the poly-A tail to wake up specific mRNAs like cyclin B, massively turning up the translational volume without transcribing a single new gene.
And we saw the ruthless, active uninstall of those maternal messages via the CCR4 knot complex and XR1 to allow the embryo to exit metaphase M2 arrest.
And finally, we witnessed the great handover. The maternal to zygotic transition, the critical major ZGA checkpoint at the 8-cell stage in humans, completely distinct from the 2-cell stage in mice. The massive ATP-dependent epigenetic unpacking of the chromatin by HTES and SWES7F complexes.
It's so much to take in.
It is, and we grounded it all in the unforgiving clinical reality. When an embryo arrests at the 2 to 8 cell stages, it is almost always screaming that there was a failure in translational control, mRNA clearance, or zygotic genome activation.
It is a stunningly complex, beautifully precarious system. You really have to marvel that it ever works at all.
It is a miracle of biological engineering, but if we connect all of this molecular theory to the bigger picture of human health, it raises a profoundly important forward-looking question. We talked extensively about how oxidative stress in the lab can immediately overwhelm proteostasis, causing rapid arrest or catastrophic aneuploidy. But consider the embryos that don't arrest. Consider the embryos that survive that stress.
Oh, that's an interesting point.
The ones we transfer.
Yeah.
Ones that become pregnancies.
Exactly. Consider how the subtle oxidative stresses we introduce in our culture systems might not cause immediate cell death, but could subtly alter the efficiency of that massive chromatin remodeling event at ZGA.
Like it just slows it down a bit.
What if the HITs and the squirrel and F complexes are just slightly impaired by a transient wave of proteotoxic stress? Maybe they don't open the embryonic promoters perfectly. Maybe a few critical metabolic genes aren't completely demethylated.
Wow.
Could a slight transient imbalance in proteostasis on day 3 create lasting epigenetic ripples? Ripples that don't stop the embryo from reaching the blastocyst stage and don't prevent a live birth, but that permanently impact the long-term metabolic health, the cardiovascular risk, or the neurological development of the adult offspring.
That is a staggering thought. The idea that a transient, seemingly minor stress on the 3D folding of a single chromatin remodeling enzyme on day 3 of culture could leave a permanent epigenetic signature on the adult human being decades later.
It really makes you think.
It really brings us back to where we started, looking down that microscope. That diagnostic textbook criteria is definitely not telling us the whole story. We are working in a murky, fluid, highly dynamic molecular landscape where the stakes literally could not be higher. Something to deeply ponder as you sit down at the bench tomorrow. Good luck to everyone taking the ESHRE exam, and no matter where you are in your embryology career, keep diving deep.
Thank you sincerely for your patient listening and dedication to mastering clinical embryology. It's professionals like you who advance our field every day. If this content added value to your practice, a simple like helps it reach more embryologists and assisted reproductive technology specialists worldwide. Please subscribe and turn on notifications so you never miss updates on lab protocols, cryopreservation tips, and guideline-driven insights. Your support fuels more free, high-quality resources for our entire assisted reproduction community. Let's grow this channel together for better patient outcomes.